Good day, fellow students. This is Prof. Tabit speaking. I want to present a lecture on understanding dependent and independent variable. The reason for this is because most of my students are struggling to understand these concepts and they are struggling to write a research objective in the proposal. So if you cannot write a proper research objective, this is only going to delay the finalization of your research proposal. Okay, so let's go into it. We basically have the dependent variables and independent variables. So how do we identify them? How do we know that this one is the dependent variable and the other one is the independent variable? How do I even know that I do have variables in my research objective? Okay, so I'm going to start with the dependent variable. The dependent variable is normally the subject that we are investigating, okay? Not necessarily the subject, the phenomenon that we are investigating. So this is it, the phenomenon under investigation, okay? Maybe you are investigating habits, you are investigating biology, maybe you are investigating bacterial quality, maybe you are investigating genetic diversity, maybe you are investigating alcohol consumption. So all those are the phenomena under investigation, because when you investigate alcohol consumption, you must have done something, then you analyze, you collect the data at the end of the day. You want to do rheology, you must set up your experiment, do your treatment, and then you make your final observation and collect data. So we do always collect the data for the dependent variable at the end of the experiment, and sometimes when the experiment is already running. Okay, so phenomenon under investigation, data will be collected during or at the end of the investigation. So that is how you figure out which one is your independent variable. Okay, the one that is a little bit more complex will be the independent variable. Okay, I have a little bit of trick to show my students on how to locate the independent variable in your objective. I mean, when you read many articles, they'll tell you that the dependent variable is the output variable and the independent variable is the input variable. Okay, but they are not going, most of the explanations are they are ambiguous. Okay, students still do not understand after reading all this. Okay, so now I'm going to explain, because the dependent variable is easy to figure out. Okay, you just imagine the experiment. Which one am I going to correct data last? When the experiment has been done, that one will be a dependent variable. So I'm talking here about experimental design. I'm going to come to survey design. Okay, right. Now let's look at the independent variable. Okay, the a little bit extra, okay, the little bit of extra material that I'm bringing in this lecture is to link the independent variable to your sample. So I'm saying that when you look at a research objective, or when you state your research objective, you must locate your sample. Okay, look at the characteristics that will categorize your sample into different groups. Okay, the characteristics that can be used to put your samples into different groups will be your independent variable. Okay, so that's the first way to note the independent variable. The second one is the different level of treatment. Okay, the different level of treatment that will be used for your samples. So if you use different treatment for the samples, you end up with groups of samples treated differently. So the whole idea of the independent variable is to categorize the samples into groups, either by inherent characteristics of the sample or by treatment, by different levels of treatment. You know, okay, I'll give you a classic example. I want to measure the amount of starch in leaves. Okay, I went to a tree and I plop leaves. Okay, so if I don't sort out the leaves into groups, it will be difficult for me to make some comparison after I've measured the amount of starch. So the amount of starch will obviously be my dependent variable. So you must look for some characteristics that can separate the leaves into groups, or that can be used to separate the leaves into groups. For instance, it could be the leaves of different color. Okay, maybe I'll go to a tree, I'll get the yellow leaves, I'll get the brown leaves, I'll get the green leaves, the light green. Then now I have a characteristics that I can use to put my samples into groups. So that color, that different color, the different colors will automatically be my independent variables. The independent variables, sorry. Okay, so those are different groups of my independent variable. Or I can get leaves that look the same, okay? I can get super green leaves. All of them look the same. They're identical, they're uniform, okay? I can even do a mixing. I can do a mixing before I select the leaves, you know? So that is a perfect sample, okay? Homogeneous sample. But I decided to treat the leaves using different levels of alcohol. So I'm using alcohol as my treatment. But now I want to use different levels of alcohol treatment. So how am I going to do that? I can have 30% alcohol, 40% alcohol, 50% alcohol. So it means I'm going to divide my samples into groups before subjecting them to the different levels. So I'll end up with samples, group of samples treated differently. So somehow I'll end up with groups of samples. So that's the independent variable. The independent variable will make you end up with groups of samples. So that is the basis for the statistic analysis, whereby you compare groups, you do ANOVA, you compare groups, you know, you can do chi-square, you compare groups, you can do other things. So what I'm saying is that if you have a research objective and the independent variable is not clear how it's going to put the samples into group, then you are in serious trouble. Okay, so that experimental design is going to be very problematic. Okay, so that's it with experimental design. So I hope you'll be able to identify your independent variable. It should be linked to samples. You should be able to categorize your samples into group, either at the level of treatment or at the level of inherent characteristics of the sample. Okay, now I move to survey design. We do survey design on humans, animals, insects. We do some survey designs on objects. Okay, we can survey vehicles, you can survey farms, you can survey cars, you know, you can do a survey in a school, you can do a survey in a hospital. Okay, so for a survey, your sample also becomes the humans or the animals or the cars or whatever. So the identification of your independent variable stays the same. There should be some characteristics that you can put your samples into groups. For instance, if I go to a hospital and I'm doing a survey on patients, maybe their recovery, okay? I want to look at their recovery or their response to particular treatment, you know? So my sample, I must have some characteristics that can put my samples into groups. So demographic characteristics can do that. Male, female, different level of education, you know? Then you also have other characteristics. It could be maybe inherent characteristics of the patient could be the kind of disease. Maybe you are suffering, one group is suffering from typhoid, the other one is suffering from malaria, the other one is suffering from maybe um, uh, parasite infection. You see, so those are inherent characteristics of the sample that we can use to put the samples into groups. Okay, so for a survey, we have only that. Okay, because for a survey study, we don't do treatments. Okay, we don't do experimental design, you don't do treatments. Okay, you don't subject samples to treatment unless you are doing the quasi-experimental design, whereby you can subject different groups to different treatments. Okay. So for those who are doing survey study, it is very clear. So if you are doing a survey on houses, it could be different. The characteristics that can categorize your house could be different roofing, different paintings, different number of rooms. You see, if it's carved, you can check. Maybe you want to check the purchase rate for a particular brand, let's say Polo Vivo. Then now you have a group of Polo Vivo cars that you sample. What is it that you're going to use to categorize the samples into groups? Okay, you can check the color, you can check the tire sizes. So you look at the inherent characteristics of the sample that you can use to put the samples into groups. So for the survey, it's very clear. Then for the surveyor, dependent variables will be the subject, the phenomenon that you're investigating. The price for the hospital, you look at the response to treatment. For consumers, you can look at maybe preference and stuff like that. So all those things that you're going to ask questions. And also remember, for a questionnaire, you can also look at checklist. For instance, you want to use compliance. You go to maybe a food service facility. You want to look at food safety compliance. You use a checklist, Are they doing this? Yes. Are they doing this? Yes. Are they doing this? So all those things that you are checking will be the phenomenon under investigation, but the inherent characteristics of the subject of the facilities will be their independent variable. And this inherent characteristic of a facility could be, do we have electricity there? Do we have potable water there? You understand? So all the inherent characteristics of the sample that you can use to put the samples into different groups will automatically be an independent variable. So I cannot overemphasize about this. It's a must know when you're writing a research proposal. Okay. So now let's look at some few examples. Okay. The first example here would be to investigate the effect of temperature on the viscosity of modified starch soups. So the first thing that we need to do is to maybe identify our sample. Okay. If we can identify our sample, then the next thing will be is to identify the independent variable by looking at the groupings, how our samples can be grouped. Is it by treatment or is it by inherent characteristics? So let's see, modified starch soup will be the sample. So I put S there. And investigate the effect of temperature. So we are looking at the effect of temperature. So temperature is the treatment, in this case, treatment. So temperature is the treatment on what viscosity. So if I can imagine this experiment, it means I'm going to have my samples, then it means I'm going to treat them with different temperature, then I'm going to collect data on viscosity. So the viscosity is the variable that will be collected at the end of the experiment. So dependent variable that will be collected at the end of the experiment. Okay, GV, sorry, GV, dependent variable. Okay, now the temperature will automatically be our independent variable, definitely independent variable temperature, because we are going to set up the experiments at different temperature. Okay, so we are going to have different levels of temperature, maybe 30, 40, and 50. Okay, so this is our independent variable. So now we've identified the sample, we've identified the treatment, and we identified the viscosity. So the treatment we meet the treatment meets the second criteria of independent variable, which is this one, different levels of treatment. Then we'll end up with samples, group of samples treated differently. Okay, so the independent variable in this objective is treatment. Okay, so now we've identified the sample, we've identified the output variable, which is our dependent variable, which is viscosity, the input variable, which is our treatment. Okay. Now let's look at the second objective, to investigate the genotypes of Salmonella species isolated from raw chicken bought from different abattoirs using MLST, multi-local sequence typing. Okay, that's the meaning of MLST, for those who are not doing molecular biology. Okay, so we need to figure out our sample. What are we going to sample? I think you can see from here we have abattoirs. We need to locate abattoirs. Okay, we need to locate abattoirs, and there are different abattoirs. We need to get chicken from there, and then we need to get isolates from there. Okay, specifically Salmonella isolates. Okay, so that's our chain of command in terms of the sample. Okay, so the isolates will carry the characteristics of raw chicken from different abattoirs. So raw chicken, it doesn't say anything, okay, whether it's different type of chicken. So it's just raw chicken. So raw chicken is constant in this case. So the thing that varies is different abattoirs. So therefore, this is what we are going to use to categorize our isolates, different abattoirs. We use to categorize our isolates. So this one meets the first characteristics of independent variable. This is it, number one, characteristics which categorizes samples into different groups prior to treatment or experiment. Okay, so here, different abattoirs will categorize our isolate. So we'll have isolate from this abattoir, from that abattoir, from this other abattoir. So it's going to categorize our isolates. Okay, maybe it's abattoir located in the city, abattoir located in the rural areas, or abattoir located wherever, but different abattoirs. So this gives me the idea that it's different. So we get isolates from different abattoirs. So this one becomes, the different abattoir becomes our independent variable, our IV. Okay. So to follow the other definition, we are going to enter the experiment with this, knowing this. So it's an input variable. Okay. So independent variable, input variable. So we need to get our samples from different abattoirs. We are going to know this thing upfront before we even start to go to our genotyping, you know. So it becomes our independent variable. So now we take the samples, we subject it to multi-local sequence typing, which is the method that we're going to use. And then what are we going to analyze? Genotypes. So maybe we'll get genotype 1, genotype 2, genotype 3. So genotype automatically will become our dependent variable, because that's we'll be collecting data from these after we've even run our MLST. So after we've run our MLST, which is the experiment, then we are going to get DV, and genotypes will be our dependent variable. So this is a classical case whereby we did not have treatment. There's no treatment here. So we have the samples. There's an inherent characteristic was used to put the samples into the category. Then we subject the samples into the experiment. Okay, so unlike the first one, the independent variable was a treatment. In the second one, the independent variable is not a treatment, it's a characteristics which categorizes the sample. So in the first one, for treatment, we'll get group of samples treated differently. In this one, we'll get different group of samples based on different inherent characteristics, which is different abattoirs. Okay. Now we move to the next one, to investigate the food safety knowledge of consumers of street vended food. Okay, this one looks like a survey research. Okay, so what are we investigating here? For survey research, you remember that we also need to start where, what is our sample? Yeah, where is our sample? Our sample will be consumers, okay? Consumers of street vended food. That will be where that will be our sample, you know? So this is our sample for survey study. I said we are looking for inherent characteristics, okay? So it will be our social demographic, okay? Social demographic characteristics of the consumers will automatically be our independent variable. So this will be our IV. Okay. Then now the social demographic will be the independent variable. So that one is clear. So therefore, the phenomenon under investigation is food safety knowledge will be our dependent variable. So like I said, for those who are doing surveys, it's very straightforward. Okay. Then for experimental research, let's look at the last one. We investigate the prevalence of nitrogen-fixing bacteria in farming soil. Okay. So sample, definitely farming soil. Okay. And then when we get our farming soil sample, we conduct our analysis. We are looking for nitrogen fixing bacteria. So we will do some kind of an identification test. So if we do the test, what are we going to record? We record prevalence. Is it there? Is it not there? Isolate one nitrogen fixing bacteria, isolate two, not. This sample, how many did we get? That other sample, how many did we get? But now I'm looking for the characteristics that will categorize my samples into groups. I can't find. Farming soil, can it categorize our soil samples into groups? No. So you see that this research objective lacks that characteristics that will put our samples into groups. So we can introduce it. We can say in farming soil from, you can see different plots, okay, from different plots, farming soil, different plots. Then we have that characteristics that can categorize our samples into group. Different plots will do that. Then we can have seasons. Maybe the soil samples, we took it from different seasons. Then it automatically becomes the characteristics that will put our samples into group. It could be from different farming soil, from different crop cultivation, whatever, where you cultivate different crops. Then that different crops will automatically categorize your samples into groups. Then that research objective will be completed. Okay, so I really hope that you've understood how to navigate around dependent and independent variable. Most of the papers will tell you that the dependent variable is the output variable, the independent variable is the input variable. Many students will memorize it. At the end of the day, they still cannot differentiate between the two. So I've tried to use the relationship between the independent variable and the sample, and the independent variables, and grouping the sample to see how you can identify your sample, look for that characteristics that can put your samples into groups prior to the experiment. It can be inherent characteristics. It can be different levels of treatment. So I really hope that you've grasped this. So I'm looking forward to the future lectures to come. I hope to see you there. All the best. Take care. And please make sure that you know how to state your research objective and also use the right dependent and independent variable. Okay, thank you very much. Bye.